This is Nature of Science Lesson 5, and in today's lesson we're going to learn how to prepare a wet mount slide, and we're going to learn how to stain a slide so that the cell parts become more visible. For those of you that haven't turned in your safety contract, it's really important that you get that in because you won't be able to participate in this lab until you do. So how do we make a wet mount slide? Well, the first thing that we're going to do is we're going to use a little pipette, which is plastic, which looks similar to a dropper, to place a drop of water in the center of the slide. On top of the drop of water, we're going to place the specimen that we're going to look at. In our case, we're probably going to look at some onion skin cells. Then we're going to place a cover slip on the slide at the edge of the water. You want to do this at about a 45-degree angle and then slowly lower the cover slip. And then you can tap with your finger on the cover slip to get out all air bubbles. The other thing that we're going to want to do is hold a piece of paper towel or a tissue at the edge of the slide to absorb any water, extra water that may have come out from under the cover slip. Stains are used to enhance cell parts and structures when we look at them through the microscope. This says recall the technique used in class. Well, we haven't used it yet. I'm introducing it now. We're going to be doing it this week.
Today I'm going to be teaching you how to stain a slide. The first thing that you want to do is you want to obtain a clean microscope slide. The second step that you're going to do is you're going to place your specimen in the center of the slide. Today what we're going to be doing is we're going to be making onion cell slides and staining the cells in onion. So what I want to do is get a very thin piece of onion skin, and once I get that thin piece of onion skin, I want to place it onto my slide and try to get it on there as flat as I possibly can. Alright, our next step after we get our specimen placed on the slide is to add a couple of drops of stain. The stain that I'm using today is called methylene blue. And what it's going to do is it's going to stain all the organelles in the onion skin here. I'm going to place a couple of drops onto my onion skin. Alright, my next step after I place that stain is I need to put a cover slip on top of my slide. Now this is a tricky part because we don't want to just put the slide or the cover slip on straight up and down. We want to come down at a 45-degree angle, and that's going to eliminate some of the air bubbles that you might get. Alright, once you do that step, you're going to tap on the top just a little bit to try to get some of that stain inside the slide. Now my last step is I'm going to remove some of that excess stain that's on the onion cell slide, and to do that I'm going to take this tissue paper right on the edge of this cover slip right here, and what that's going to do is it's going to draw the stain through the specimen, and it's going to stain every one of those little cells that are on my slide. And this part, sometimes you do get some stain on your fingers, but that's not such a bad thing. It just shows you've been doing some biology. And I'm just going to clean up this side of the slide. And then once you're finished, you have a stain slide, and my cells are going to show up nice and bright on that microscope. And then the very last step would actually be to view your slide on the microscope. And it looks great. I've got a lot of nice darkly stained cells on my slide. Thank you.
Okay, so that's what we're going to be doing in class in our lab. Last two things I want to explain is what is gel electrophoresis. Gel electrophoresis is a process that's used to separate mixtures of large molecules according to size. It's most often used to separate DNA. Electric current pulls the bands of the DNA through the gel and it creates a pattern. And you'll read something also this week about how this is used in forensics when they get a sample of DNA and they have different suspects of a crime. They can match up the DNA of the suspect to DNA that may have been found at a crime scene. And lastly, chromatography. You may have done this in 7th grade life science, where you use a piece of chromatography paper and you can use it to separate pigments in a mixture based on their density. The less dense molecules will move faster and further to the top of the paper, where those that have a greater density will stay toward the bottom. And we can do this with ink and different pigments that are found in different substances. And that's it. Very short lesson, and we only have one lesson left in this unit, and then we will have a unit exam.